Thank you, Wingong. Thank you so much for giving me this opportunity and inviting me and Charles to share some of the work that we've been doing in the past 2 years. So my name is Jen. I was a postdoc actually here at the Broad, and so I would like to just start with my acknowledgement slide. So I did my postdoc with Eric Lander a few years ago, and also co-mentored by Rudolf Jenisch. And many of the work that I'm going to talk about today is actually in close collaboration with Aviv, and also Charles, who's sitting there, is my first PhD student, but it's also co-mentored by previously Aviv, now with Colin now. So that's why I would like to start with a thank you first, because without their help, none of us would be here. And then also would like to start with thank you for the team, because I was a junior faculty. I started my lab 2 years ago at MGH, and these people are very brave, and then they just joined my lab, and then they've been working on very challenging problems related to how do we generate omics information from different kinds of imaging modalities. And also would like to thank all the funding agencies in the past few years, so that none of this, without this, we can't do anything. So really want to thank them first. So, and the big questions that we're interested in the lab are divided by decoding, modeling, and reprogramming, 3 categories. So how do we actually use these different approaches to decode, model, and reprogram cell fates and cell circuits? So from the decoding part, that we were interested in developing new single-cell spatial genomics and especially imaging technologies to understand and map the cell fates and circuits. So that include a wonderful collaboration with Aviv that we developed Raman to RNA, which is an experimental and computational framework how we can actually infer single-cell RNA-seq data from Raman microscopy images. And then I think it's going to be on today on bioarcs that SHAF that Charles has worked with, also again with Aviv, that he developed this machine learning tool to infer single-cell gene expression from histology images. And then moving forward, we're interested in how we can expand what we've been doing in the past 2 years to other imaging modalities, and which are not quite popular in the biology world yet. And then we're also interested in developing new tools to map not just the host RNAs, but also the microbial world, the bacterial virus in tissues, and also the mutations, how we can actually map the point mutations in tissues. So that's just the first part. How can we actually decode and map the gene circuits and cell fates? And the second part is once we have this, mapping tools, how do we actually study a complex molecular or like a cellular system? So we need to have some better organoid systems. So the 2 tissues that we're interested in are the skin organoid and the placenta organoid. But today I'm not going to talk about the organoid work. And then the third part is once we, figure out ways to decode the gene circuits and then the cell fates, and we have a model system in vitro to really model this, how do we actually reprogram the cell fates and cell circuits to allow the cells to perform new functions? So that's why we're interested in all sorts of reprogramming, lineage reprogramming experiments too. But with that, we would like to use some of this new, single-cell sequencing technologies and imaging-based, also like a phenotyping approach to do this kind of large-scale screen to find new reprogramming strategies. So that's the high-level overview of what we're doing in the lab. And also, like for today's talk for the primer, I would just focus on very high-level conceptual directions to how we can think about these problems. And then I'll leave the details to Charles. So for today, I will talk about how do we decode the time information, how do we basically rebuild the time machine. And then the second one is in terms of how do we decode the cell fates. Once we figure out how to reconstruct time, and at each time point, how do we map the phenotype. And then the third, very briefly, we will talk about how can we use some of these imaging technologies to reprogram the time and phenotype information. So the first one, very brief, how do we actually reconstruct time information during the development process or any disease progression process? This was what work that when I was still a postdoc with Eric, and I think some of you might have heard of this, that the big challenges in the single-cell gene expression, sequencing is that how do we actually reconstruct the developmental trajectories over time? And there are many different other ways to, reconstruct trajectories. And at that time, most of the trajectory inference methods are more, I would say, like a deterministic, but like in development process, it's more like a stochastic process. So we wonder, how do we actually map the distribution change over time? And it turns out that optimal transport was actually a good way, like a very old mathematical concept that can be applied to really solve this problem. So the model is that if you think about each gene expression profile, it's just a sample from a time varying, like a probabilistic distribution. And then the goal here is to actually minimize and maximize the probability distribution change over time. So we apply this to study our favorite system, which is the iPSC reprogramming system. So we collect the single-cell RNA-seq along a 18 days developmental, like a 18 days experiment assay, and then we did single-cell RNA-seq and generated by the time probably the largest single-cell RNA-seq data for reprogramming. And then we use this optimal transport framework to develop basically a Google map of the reprogramming process. With optimal transport, you can predict what is the past stage, and also you can predict what is the future cell phase. And this is a beautiful collaboration with Aviva Eric. And then as you can see that with optimal transport, you can basically build this time machine that how do you, watch where these iPS cells are from in earlier time points. You can also do this like reverse, how you can predict if you have like some fibroblasts, where the future state might look like in the next time point. So that's just like a very brief intro of how do we use some of the single-cell sequencing method to decode the time information and to rebuild this time machine in development. And what we are working on now is to expand optimal transport framework into spatial transcriptomics, and how do we use this to infer the spatial trajectory. And Kodaki, who's not in the audience today, that he developed this new method called SWOT, Spatial Waddington OT. And then we're also working on how do we use optimal transport to integrate different kinds of data modalities. And then the third one that we just started to work on this is to use this to build the models to predict the drug perturbations from screening. So that's a very brief introduction about how can we use single-cell sequencing to reconstruct the time information. And then, which we will, then we move to the second part, which is something that we're working on now. And then also, this is very early stage. I would like to, seek more feedback, is how do we think about, rethink about like a phenotype. So the grand challenge, or like a more philosophical question here, is that how do we actually define what is a cell type, what is a phenotype, what is a function of a cell? And originally the cells were discovered by microscopy. So you look at under the microscope, microscope and see, oh, this cell looks like this, and then they have different morphologies, and then this is a neuron, that is fibroblast. So it was defined by morphology through microscopy. And with the recent event, with the single-cell sequencing technology, and such as the Human Cell Atlas that Aviva and Sarah led this international effort to map all the single cells across the body using single-cell sequencing technologies to define what is a cell type and what is a phenotype. But there are big challenges for sequencing. First of all, it's still very expensive, and it's kind of like a luxury game that like sometimes it depends on how much funding you have and how many cells you can sequence, and then you got different competitive advantages over that. So first of all, it's still very expensive, although the cost per cell dropped significantly, but it's still a quite expensive experiment to run. And then the second one, I think it is the biggest problem for all kinds of sequencing-based phenotyping approach, is that it's destructive. So for all these kinds of sequencing methods, you have to kill the cells to get the information from the cells. So once you kill the cells, you won't have the same cell in the next time point, or you cannot do this with humans, or you cannot do this with in vivo settings. So I think that's the biggest challenge for genomics, that this is a destructive measurement. And then the third one is, it's still, you can do millions and millions of single cell right now, but it's still a very low throughput process, thinking about how many cells we have in just one human. And if you're thinking about expand this to the entire, animal kingdoms, or like, the species and the microbes in each human, that we need like much much higher throughput measurement approach. So that's why we think about, what are the other, phenotyping approaches? Then we come to this very old, actually, way to define a cell through imaging. But there are a lot of advantages of doing imaging. So first of all, it's low cost. The running cost of many of these imaging methods that we're testing now is basically the electricity bill. So constantly running this instrument. And then the second one, I think it is the biggest advantage of doing imaging, is that it can be non-destructive, which means that you can apply this to study, for example, human samples. You can engineer endoscopy and put it in human, and then also you can track this over time. You don't need to sacrifice the cell. You don't need to kill the cell. So this will give you the time information, and it will also allow you to really monitor live cells and in tissues and humans. And the third one is that it can be very, very high throughput. With automations, you can, image millions or billions of cells in relatively manageable cost. So that's why we think, how can we actually combine the best part of this to, phenotyping approach? And then, and that's why we started to think about, can we actually just link the imaging, different kinds of imaging data with different kinds of single cell omics data? And the imaging data, it can be, morphology data, it can be chemical composition, it can also be the fluorescent data that you can generate from imaging. And then the genomics data could be, transcriptome, proteome, and metabolite information. And then that's why we propose this Image-to-omics framework. But it's not a new problem for machine learning field. It's a classic, multimodal and multi-domain translation problems in machine learning. And with this, will enable us to do very low cost and scalable mapping of genomics information in tissues and humans. And then with imaging, will also allow us to do this, to track the time information in vitro and in vivo settings. And I hope that this could be, like an ecosystem and a database for the development of new ML technologies and then also genomics tools. So the technology foundation that will enable us to really do this image-to-omics framework is actually from the machine learning. And with the recent advent, especially many years ago, Dr. Fei-Fei Li at Stanford, by the time at Princeton, they put together this ImageNet, which revolutionized the computer vision. And then more recently, the AlphaFold from DeepMind that also did this very cool work to generate the protein structures. And even more recently, OpenAI is very popular, that everybody's using ChatGPT. Before ChatGPT, that they released this DALL-E framework to generate image data from text. But all of this share one thing in common. They're doing a translation problem that we are trying to do. So for example, in the ImageNet, that they were trying to translate the imaging data, the imaging information into the labels. And from AlphaFold, they're trying to translate or generate the structure from the sequence information. And then for DALL-E, they're trying to translate the text information into imaging data. But, for example, AlphaFold and DALL-E, both the sequence and the structure are describing the same object, the same information. And the text and the imaging are also describing the same, information. That's why we started to ask, can we actually translate the imaging data into all kinds of omics data? But before that, there's also lots of foundational work that people have been doing in the field. First, in imaging to imaging. So how do you translate one type of imaging data to another type of imaging data? This work was done by Google and Stephen at Gladstone at UCSF. That they developed this method called in silico labeling using a deep neural network to generate the unlabeled fluorescent image, to generate fluorescent labels from unlabeled images. So this is to translate the bright field images into fluorescent imaging labels. So that's image to image. And then there's also lots of work related to image augmentation, also like using low resolution imaging data to generate high resolution, even super resolution images. So this work, they developed a method using GAN to generate high resolution fluorescent microscopy data from just regular low resolution data. And I think recently the bioRxiv paper from Yale University, they actually used this regular microscope, bright field microscope combined with expansion microscopy, and then some of these neural networks to generate a super resolution images just from low resolution bright field images. So this is a very exciting time that we think about how do we combine this kind of machine learning tools to really scale up the tissue mapping effort. And then that comes to a more clinical relevant studies by this group. I think it's actually a company now that developed this HE to RNA framework that you can predict the whole slide at the whole slide level, that you can predict the RNA-seq data from HE staining. So that was actually a foundation that why we were actually started to think about like Charles's work, that how do you actually generate the single-cell RNA-seq data from HE staining. But this work was mostly at the bulk level. And then Jen Doe's lab at the Stanford University, they also developed this STNet that you can generate single-cell gene expression around like 100 genes from connected Visium data, which is lower resolution at a 100 micron level, that you can train a model to, they use a CNN model to generate the spatial transcriptomics data from paired HE staining. But this is not at a single cell resolution yet. It's at the tile level. So each tile contains multiple different cells. So, and then a beautiful work and a framework, actually a general framework developed by Carolina Wooler at the Broad and MIT, that she proposed this autoencoder framework to basically compress all kinds of different input data, and then reduce the representation in the latent space, and then connect those representation in the latent space, and then decode the all kinds of output of interest. So, for example, this one they use the DAPI stainings to generate the single cell RNA-seq data, and that's more of how do we generate different kinds of omics information or imaging information from other data modalities. And then there are also some other very cool technologies that they focus more on the integration. So how do you integrate different kinds of imaging data with different kinds of spatial transcriptomics data? For example, this beautiful work from Oz lab, and that they were trying to integrate the MERFISH data with electron microscopy images. So you can have very high resolution morphological features from the electron microscopy. At the same time, you can also have high resolution gene expression data. And also like here, for example, at the Broad and Carpenter's lab that they've been doing, they pioneered the cell painting and for drug discoveries, that we're also trying to integrate the cell painting data with L1000 gene expression data, and then how you can just basically infer the gene expression from this cell painting information. So that's basically all the background that I wanted to give you about different kinds of imaging and omics data that people have been trying to generate and link them together. But here today, then I think Charles will talk a little bit about in detail is how do we think about other imaging methods beyond just traditional fluorescent or bright field imaging. That's why we were thinking about how do we link new imaging modalities with all kinds of omics data. But specifically, the imaging method that we are interested in are it's called chemical imaging. It's a very high dimensional measurement, which is not quite popular now yet in biology, but it measures super high resolution and then also high dimensional information per pixel. This one, for example, can give us up to 1,000 dimensions per pixel. So, and the first work, it was a wonderful collaboration with a former postdoc, Kazaki, in Aviv's lab. And then when we published the Waddington OT paper, and Kazaki just started at the Broad with Aviv, and then we started to chat, and then can we actually use some of his Raman microscopy images, and then infer the single-cell RNA-seq data that we generated from the Waddington OT manuscript, and then we developed this method called Raman to RNA, and this is also a collaboration with Peter So at MIT, because he has a center of all kinds of very advanced imaging instruments at MIT. So this one is how do we actually generate the RNA-seq data from Raman microscopy, but what is the Raman microscopy? What is the chemical image? And at least this is not an imaging method that we would usually see in a single-cell gene, single-cell sequencing talk. A chemical imaging method is type of it's a new category of imaging methods. Raman is just one type of imaging, chemical imaging method. But what Raman does is it measures the chemical vibration inside of a cell. So each molecule has a vibration, has different vibrational frequency. And then you shoot a laser to the samples, and then different molecules, they are vibrating, and the light will be scattered in a different way. And then you capture the scattered light, and then you will get like a spectrum per pixel. But with this spectrum, it's high dimensional, up to 1,000 peaks. Think about fluorescent, you have 5, but this has 1,000. And then it's a composite measurement of every molecule inside of cells versus we're measuring RNA. You are measuring lipid, proteins, nucleic acid, and carbohydrate inside of a cell all in once. But it's composite measurement. We're not measuring each individual molecule. So if you're asking me like, oh, which protein is actually this, we don't know. But there are computational ways to convolute all this information too. But like for this, we're doing a composite measurement of different kinds of molecules inside of a cell. So this will give us a fingerprint or like a profile of a cell in a non-destructive and label-free manner. Just put the cell there, take a picture. They will give you 1,000 dimensions per pixel. And another work that will be out today on BioArc is Charles's work called SHAF, that he developed used like adversarial autoencoder framework to generate a single-cell RNA-seq data from the histology images. And Xinjian, who is another a researcher in the lab, that he also developed a similar approach to generate single-cell RNA-seq from histology images when he was a student in grad school in Hong Kong. So that's just two imaging methods that we tested. So one is the Raman microscopy, and then the second one is histology. And then beyond just the Raman and the RNA-seq, we're testing different kinds of other chemical imaging methods. For example, this one on the left is that we generate this images of a mouse brain without any staining, and just cut the mouse brain, put it under microscope. But this one at the subcellular resolution, it only took us like 10 minutes to scan the whole mouse brain. And then we're doing mouse. Yeah.
Not necessarily pixel-wise accurate, or like, how does that factor into the downstream application from
so this is basically, I think it's still at the very early stage whether we can generate omics information from imaging. I think the results are more qualitative versus quantitative, but I think this is basically a new direction that we're thinking about phenotyping. Think about like AlphaFold like 10 years ago, and you have to do structural biology to really get the protein structure. And those true here, right? We have to do sequencing to get gene expression. But actually, gene expression is just a 20,000 dimension vector, and it's not that big. It's big, but it's not crazy. So that's why I think we're at the very early stage of thinking about whether this is feasible and what is the right imaging methods we need to use. Yeah.
Sorry, can I ask a follow-up then?
Yeah.
So like, I guess in the example you gave, we know that structure is completely contained in the sequence, but in some of these examples, it's not clear a priori that all of the, say, structure information is captured in the image.
Yeah.
It's like, how do you think about that, or how do you handle that?
That's a wonderful question, because we don't actually know because we know the output, right? We want the gene expression as the output, but we actually don't know what is the right input. We tried Raman, we tried HE, because each image method, they capture different kinds of features of the cell state, right? For example, the Raman microscopy, they capture the chemical information inside the cell, and histology, they capture, morphology information. But thinking about, the causality here, actually, the gene expression will be the upstream, and then the result of a gene expression, program, the cells will have different morphology. They will have different kinds of molecules inside the cell. So we're measuring some of those features, but we don't know what is the right combination of different kinds of imaging measurements right now. That's why we're testing, different kinds of imaging methods. We also wanted to really think about, which I will talk in the next few slides, speed and resolution, and how do you really thinking about the advantage of doing imaging. If it's like super slow, then probably we just do sequencing. But I just wanted to highlight the advantage of some of this imaging method that we're testing now. You can really, for the first time, to do this at a single cell resolution at whole organ level. We just imaged like a monkey brain, which took us like 1 hour, and then we're moving aggressively to try to see if we can map like human organ in a few days, not like years, right? So that's like, beyond Raman and single cell RNA-seq, what kind of other imaging methods we can try, and what kind of other single cell sequencing methods we can thinking about to link them together. And another way that we're thinking about this is all the imaging methods that we're talking about, those are like label-free imaging methods. You don't need to do any staining. Once you do the staining, you will have some perturbations to the system. And then, but as a compromise, we're also thinking about how do we scale up just with labels. So there are, for example, if we're using fluorescent labels, there's 5 different colors. And sometimes if you use, some fancier microscope, we can push probably 8 different colors. And this is a beautiful collaboration with Wei-Ning at Columbia University, who's a pioneer in chemical imaging. He co-developed an SRS, which is one of the faster versions of Raman microscopy. And then they also pioneered the work to use chemical dyes to replace the fluorescent dye and then scale up labeled measurements. So for example, with some of these chemical dyes that they developed, you can do up to 200 different colors. This is like not the fluorescent color, but like colors that can be measured or mapped by chemical imaging methods. So if you really come back to think about the advantages of doing chemical imaging with labeled and unlabel-free methods, you think about like the labeled dyes, fluorescent imaging might allow you to do maybe 4 or 5 different colors. Chemical imaging can allow you to do up to 200 different dyes. Of course, there's a lot of challenges here. The signals is not strong enough, but people are working on to make it stronger and stronger signals. And then if you think about like the label-free approach, then phase imaging probably give you like 3 different, dimensions per pixel, and then fluorescent give you maybe like again 4 to 8, on average maybe 5 different colors. Chemical imaging at the pixel level will give you up to 1,000 dimension information. So it's 200 times more information you can get compared to traditional fluorescent microscopy. So if you really think about it, how do we really do this phenotyping? There's imaging-based phenotyping approach can be very scalable. And at the same time that you can also do single cell sequencing-based phenotyping approach, you will get a lot of information about the cell state. But we're thinking about how do we combine this two so that you can achieve both the scalability, and at the same time, you're going to generate a lot of information about the cell state. So if you really think about like the translational opportunities that Image-to-omics can be applied to develop new science tools and machine learning tools and an omics platform for tissue mapping. But we can also think about like the translational opportunities in drug discovery, drug screens. How do you use some of these chemical imaging methods for very large scale perturbation screens? And then you can also use this for diagnosis purpose. For example, that you just take a picture of a cell and then you infer the omics information from that cell. Then you know whether this cell is, a good cell or a bad cell. Because for some of this, cell therapy or the cells that's very special or like rare, you cannot do sequencing, because if you do sequencing, the cells are gone. You can't use that cell anymore. So we need non-destructive ways to phenotype the cell and then keep the cells alive. And then that's basically the first two main parts. And then the third part, I would just touch base very briefly how we can use some of this, approach that we just mentioned to reprogram the time information and then the phenotype at each time information. So that's what we are very interested in, reprogram the cell phase. Just a little bit background introduction about the reprogramming that Dr. Shinya Yamanaka developed the first protocol to make iPS cells in 2006. And then in my PhD, I proposed this T-cell model in 2013 that we developed like a new cocktails to make iPS cells that you can make stem cells with just differentiation factors, not just the stem cell factors that people are using to make stem cells. So this is the high level introduction about what is this T-cell model. So what we found and also proposed is that all these different stem cell factors, their function is to actually drive the cells into differentiation. So it is the balance between different immune-specifying forces. That's why the cells can be maintained in the stem cell stage. And then after our model, then several other groups found that this is actually the case in many other systems, such as neural systems too. And then if you follow tech news recently, you probably found that many of these companies in the past, come up in the past 2-3 years, they really are focusing on the cellular reprogramming for longevity and anti-aging research. And the foundational principles among all these new efforts to push the longevity is that how we can actually just reprogram the time information during development, which is something that we talked about in the first part, and how do we use some of the new measurements to really do this. So there are just like examples of 3 new startups that using some of these reprogramming strategies to reprogram the time of a cell. I mentioned that Waddington OT manuscript, like a paper that we use single cell RNA-seq based phenotyping approach and to find new reprogramming strategies. And from this work, we actually found one new factor called GDF9 that you can actually significantly increase the reprogramming strategy. But combining some of this new imaging method that we just talked about, how we can, for now, we're working on how do we use some of these chemical imaging methods combined with perturbations to do very large scale screens to just to search the whole genetic perturbation design space. How do we do combinatorial screens and then find new reprogramming strategies? So in summary, then we talked about how do we use optimal transport to decode the time information, and then how do we use Image-to-omics framework to understand the phenotype of a cell, and then because time graph phenotype is actually videos of cell states, and then for now you can also just generate the videos from text, and then can we generate a developmental videos from the gene expression measurement too. And then the third one is that we give you like a very brief example. How do we use the reprogramming strategies to reprogram both time and phenotype information? But where are we now? Then we talked about, we wanted to do this, Image-to-omics, which is very challenging and we can do it alone. So where are we now? So I think we're still trying to figure out what kind of imaging methods are more appropriate to generate omics information. And then another challenge is that there are very few paired data. There's very limited amount of data that's paired. And then there's also not very organized in a way that you can do the ImageNet challenge. And then also there's not much perturbation data that you can really apply to do this. So these are the challenges. But step by step that we can work on some of them. And for now, we're working on and then exploring what kind of imaging data are the right input for us to generate the omics information. So we're thinking about we started with the Raman, we started with HE, and then what are the other imaging data that can be potentially used for generating the gene expression as an output, right? So a little bit some thoughts that we have here is that in terms of label-free and multiplex labels, so we're trying different kinds of label-free imaging methods, and we're also trying multiplex the chemical dyes at the same time. And then I think it is very critical to think about multimodal, that we wanted to measure different kinds of features from the whole feature space, not just in the similar space. For example, all kinds of multi-omics measurements that are measuring similar features across the whole feature space that represents like a cell state. So we wanted to measure orthogonal features from the same sample. For example, can we do imaging, spatial omics, and chemical imaging from the same sample? So that's some people are working on that in the lab. And we also have to think about the dimensionalities and all of this work. Those are still at the stage are qualitative because the limitations of these imaging methods usually are very low dimension. But gene expression is a high dimensional data. How can we generate high dimensional imaging data? That's something that we are also thinking about. And then the fourth one is actually the speed, because some of these imaging methods, they're great, they're non-destructive, but they're just so slow. So we have to really balance, if it is even like the advantage of doing imaging, if it is too slow, then probably we just go for sequencing. And then the last one, I think it is the resolution challenges. But for some of this very high speed imaging methods, we have to sacrifice the resolution to achieve the speed. So what we're working on now, I think Charles and Xinjian and some other people in the lab are working on, organize different kinds of image-to-omics approach into a more generalizable framework so that we can, really achieve image-to-omics. And then people are also doing, if we can generate omics information from images, can we actually do the reverse? Can we generate imaging data from just omics data? Can we generate, for example, spatial transcriptomics data just from single RNA-seq without the spatial information without the imaging? Can we actually generate that? And then that's why, like from the experimental side, lots of people are in the world in the lab are actually generating this kind of paired data. And Charles and Xinjian, they are also doing a survey on what kind of publicly available data sets are available now. And so I think in summary, this is a long-term vision. We can't do it alone. So AlphaFold basically they're trying to generate the protein structures from the sequence. And in the OpenAI study, they're trying to generate images from text. We asked the question, can we generate all kinds of omics information from different kinds of imaging data as an input? So if you're really thinking about the AIGC, like AI generated content, which is popular now, can we propose in the future to do, AI generated cells? And AlphaFold and Rosetta from David Baker, they were trying to do, de novo design of proteins. Can we do de novo design of gene expression or gene regulation programs in the future? So by the last slide is basically a thank you to summarize all the work. And this is a team effort for sure. We started from 1 people 2 years ago, and then we have like a decent amount of people in the lab now. But we're also hiring. Anyone are interested, just let us know. So thank you.
Hi, everybody. I'm Charles. I'm a PhD student. Wengang just told you this. And I would like to talk about, specifically inferring transcriptomic information at single cell resolution from H&E stains, but also a little more broadly about what my PI John was talking about before, this full concept of, a large scale framework for inferring omics from images. So first, this is a brief set the stage. More or less, there are a lot of downstream tasks that biologists, biomedical people aim to answer, drug screening, diagnostics, medical professionals, and a lot of them can be reduced to just this very simple question of what do omics data look like. Proteomics, we're very interested in transcriptomics, and the whole family of data, that is around this. Now, the issue with this, I'm sure everybody is very familiar. These data are very expensive to produce. They're very powerful. They give incredible vantage points into biological systems, but require very expensive instrumentation, a lot of time, expertise with really idiosyncratic laboratory materials. So there's a lot of pretty much cost limiting factors with actually obtaining this data. This being said, we'd like to consider, biomedical biological imaging as an alternative, spanning a whole range of different modalities, whether this be more traditional biomedical modalities such as H&E stains or DAPI stains or whatever have you. We can look at our friends in the physics department and look at more sophisticated spectroscopic modalities, whether it be Raman spectroscopy, as John was talking about before, or SRS or whether have you, or even, what we'd like to see as a very long-term goal is looking at really more canonical medical images like mammograms, MRIs, et cetera, and see if we can, notice that, these are a lot easier to be produced than omics data. It's a lot faster with less equipment, much more universally available. And we'd like to have a question more or less, how can images complement omics data? How can they not really replace, but really enhance and maybe take away some of the burden of having to produce omics data? With this being said, as with anything in data-driven sciences or machine learning, we have to look at what is the connection between these two. And more or less, we'd like to think that there is a shared signal between a tissue's imaging data and its omics data. More or less, this is kind of, I think this intuitively, biomolecules of interest. Really, they manifest different phenotypes of the tissue in certain ways, like pathology look like, what does different intensities at different point of the images look like. And more or less, we'd like to think that there is a shared signal between the two. And because of that shared signal, we can exploit that and ask the question of, can we use data-driven models to connect the two? And when I say connect, move from one to the other. This being said, we can also think to our friends of the computer vision community. Really, computer vision made a really large breakthroughs in the early 2000s, early 2010s, late 2000s, with the presence of a very large comprehensive data set. In this case, we have ImageNet, as John was talking about before. This is Fei-Fei Li's, signature work. And more or less after this, we saw CNN really took off, AlexNet, people really start paying attention to this kind of stuff. And we see a similar pattern in biology too. I don't have the graphic with me, but we've all seen that graphic of, protein database introduced, and then we have AlphaFold, this database introduced, have this, ML breakthrough. So what we'd like to do is more or less, see this as a two-pronged approach. We have experimentalists and we have biologists interested in cultivating, such a large database. And we also then have computationalists, such as myself, that are interested in exploiting said data, informing biologists what data they should produce next, having a constant feedback cycle, et cetera, et cetera. But underpinning a lot of this computational work is more or less a very simple research goal. And that is that we'd like to build models that infer tissues, omics data. In this case, we're interested in transcriptomics. And we would like to do, generate data that is spatially accurate. That is an incredibly important thing. There's a lot of the information they're interested biologically is more or less spatially motivated, like what parts of the tissue, what is the cell-to-cell communication, et cetera, et cetera. And, that information is lost if we look at single-cell RNA sequencing. Of course, bulk RNA sequencing doesn't have this. We don't really have that information. It gives rise to spatial transcriptomics, et cetera, et cetera. So we'd like to, see spatial information in a tissue's transcriptomic data. We'd also like to see single cell definition. More or less, this allows us to define cell types. Very obvious reason that you'd want to have high resolution in this respect. And we'd like to get this from imaging data that is as non-destructive as possible. I was going to say one of the real drawbacks I was going to say, as John was alluding to earlier. Also, John, if I'm saying anything that's inaccurate, feel free to raise your hand and say that's wrong. But one of the real drawbacks of a lot of the sequencing technologies is that they are inherently destructive. You will have, a tissue and you will do sequencing and the tissue is gone. It's very sad. And we'd like to ideally not have that. And ideally, we'd also like to do things without, spatial transcriptomic answer. This is a little bit more of a wish list type thing, more or less because, as John was alluding to before, Caroline made this very elegant framework of how to do adapted RNA or broadly from one modality to the other without having to have really strongly paired data. Like I was going to say, the model that I'll be discussing, primary focus of this talk, is more or less just dependent on having histology data and single cell data, what I like to think of as weakly, so from the same patient, but we don't know, okay, this cell corresponds to this point of H&E stain. That is the kind of thing that we'd like to continue doing ideally. That might not be feasible in all cases, but ideally. And with that being said, before I continue working, this then sets the stage for, okay, we'd like to go from images to omics. We have this very broad goal. And we have to think, okay, what kind of images should we work with? Again, as I said before, there's pretty much these three families that we're interested in. Is legacy biomedical, let's call it, things like H&E, things like DAPI, spectroscopic, Raman, that's a fancy stuff, SRS, that's fancy, PIR, that's fancy, and clinical data, which again, that's a real wish list type thing because that's the hardest probably to learn from, but it's a nice thing to keep in mind. And for the purposes of this talk, we'll be working, largely with H&E data, so we'll start with that. Each of these three families has their merits and disadvantages, and without further ado, let's start talking about the main subject of this talk. As some background for those that are not familiar, Hematoxylin-and-Eosin images. These are very well established, very widely used in the biomedical community, the pathological community. Imaging method that more or less gives the key takeaways from this is that it's very, you get to see a very close up what a tissue looks like. As you can see here, you get to see it, very keen details of single cell morphology, et cetera, et cetera. And more or less what this gives you is just like real insights into what tissue's morphological information, how it presents itself. And then single cell RNA sequencing, more or less, is, again, we're all very familiar with this, but just very briefly, this is more or less you have a tissue. It's broken down into tens of thousands of individual cells. And for each of those cells, we have, expression information for approximately 20,000 genes. So that'll just tell you, okay, how strongly is this gene expressed? How strongly is that gene expressed based on transcriptomic data? There are a few drawbacks to it in terms of data quality. It's quite noisy, very zero inflated. It's destructive, incredibly expensive. But at the end of the day, you get single cell resolution transcriptomic data, and that's pretty great. And with this being said, this gives rise to the work that I'll be talking about. This is the single cell omics from Histology Analysis Framework. Not as catchy as SHAF, so we call it SHAF. And this is going to be on the bioRxiv imminently. We submitted it yesterday to the bioRxiv. They have a whole, make sure that there's no, weird stuff in the manuscript before they post it. So they're making sure of that. It takes about 48 hours. Excuse me. It's going to be on the bioRxiv soon. It's a submission thereafter. Part of the Human Cell Atlas with Reprogramming and a lot of papers that are being involved with that. And more or less what we're trying to do here is that we will have a histology stain, and we build machine learning models. And from those machine learning models, we'll be able to infer from this histology stain transcriptomic data at close to single cell resolution with relatively nice spatial resolution and with this high throughput. So what I mean by high throughput is that for those that are not familiar, if we have a tissue and we want to do spatial transcriptomic, if you want to perform an assay that gets you spatially resolved transcriptomic information, there's methods like MERFISH, StarMap, very, very exciting, popular technology. A lot of the times you will only be able to, because of the fact that it's incredibly expensive, only get maybe 900 to 1,000 genes, much lower throughput than single cell RNA sequencing. So one of the cool things about this method is that we do still get 20,000 genes worth of information for each cell. And in terms of like just what this looks like, for example, at the tissue, what we can do is more or less put it through our model, and then annotate the cell types of the predicted transcriptomic data from our model, and you get a map that looks like that. Very pretty. And with this being said, I'll begin actually talking about the work. And before we continue, our data more or less looks like this. We have an odd setup, in a lot of these types of settings, particularly supervised learning settings. You'll just have, okay, we have 20,000 data points. They have 20,000 labels. We're gonna take like a training set that has 13,000 data points, a validation set that has 3,000, et cetera, et cetera. And what we have here is more or less, a corpus. And we'll consider a corpus, and that corpus will maybe have 12 patients. And for each of those patients, we'll have 2 modalities worth of data. You're going to have an H&E stain of a tissue of interest, and then you're going to have a corresponding single cell data set. So for each of these 12 patients, you'll have a data set of 20,000 single cells information, then you'll have an H&E stain. And you'll have 12 of those, and then the objective is going to be more or less to learn a model that will go from the H&E stain to the transcriptomic data. So anyway, we have 3 of these separate corpora that we will showcase the method on. The first of which is from lung cancer data from Sloan Kettering in New York. I believe it's about 24 patients we have. Then we have 2 metastatic breast cancer corpora, both from the Human Tumor Atlas Project, one of which is from single cell RNA sequencing, and one of which is single nucleus RNA sequencing, which is, for those that are not familiar, just 2 different methods of going about this: single cell granularity, transcriptomic measurements. And again, we consider all 3 of these corpora to show that more or less the model is robust to different types of data settings. Lung cancer has approximately 24 patients. I believe that both of the metastatic breast cancer corpora has roughly 8 patients. And again, so that's for data setup. And our actual model is, the first thing we're going to do is we have a histology stain like we see here. And we'd like to first get more or less these kind of single cell-ish types of representations from these H&E images. So what we're going to do is actually just tile our H&E images. It's a very popular technique. A lot of other histopathology type analysis papers usually start with this, but we're just going to slide a window across our histology stains and tile them so that each tile will more or less represent, in theory, a cell in its surrounding neighborhood. And then this going forward, what we now have is, for a corpus, a lot of tiled H&E images and a lot of single cell RNA sequencing data. And we are going to actually leverage the framework that John mentioned from Caroline Uhler's lab to learn pretty much a common representation, latent representation of these 2 modalities. So we're going to learn 2 autoencoders, the first of which is going to take in H&E tiles, each of which representing roughly a cell, going to encode and then decode, very straightforward. You have another autoencoder that's going to do the same thing with the single cell data, encode and decode. And the caveat here is that you're going to have that they encode to the same latent space. So more or less, how that's done is that you'll have one autoencoder that just learns to reconstruct, and then you'll have another one that learns to reconstruct, and additionally is kind of like bullied into encoding to the same latent space as the first one. And then to actually do, the transfer, we now have these 2 models that encode to a similar latent space. It's great. If you want to go then from one of these H&E tiles to a transcriptomic profile, you can take the encoder of one model. You can encode, an H&E tile. You take that encoding, you pass it into the decoder of the other model. Now you have a transcriptomic profile. It's pretty cool. And again, they have the same latent space. So it should work. I hope it works. And after that, we have very nice output. Now, before I continue, I just said a lot of words. So if anybody has any questions, be happy to maybe take a question or 2 before going on. Yes.
When you're doing the tiling, are you purposely making sure that there's one cell in each tile, or is it just a random one?
Excellent question. So in order to set up the stage for future work, because it would be great if we segmented the cell and we did all this stuff to make it better, we wouldn't be able to do that if we just did that initially. I'm just kidding. But with that in mind, we actually, the cool thing is we kind of just ran the first experiment and we just did it, just sliding a tile, not really thinking about segmenting or anything like that. And the results were actually pretty nice. So we said, oh, this is cool. We should keep doing this. And so with that being said, in this iteration of the work, we do not do that. Okay, no other questions. I assume everybody knows exactly what is going on. So with that being said, we talked about our output. Our output is, again, spatially resolved to an extent. Like you have a tile, you have the transcriptomic profile, so you know that tile corresponds to that profile. It is spatially resolved. It's nice. High throughput. Each of these profiles you generate roughly 20,000 genes. And you know, H&E, you know, it is by definition destructive. I think we can agree it is less destructive than single cell RNA sequencing. So all big wins, if you ask me, very big wins. And in terms of how well do we do? So before I continue, this is our first, you know, kind of like the method works. And what we do is for each of these corpora, we'll roughly divide it so that we have 75% of the samples will be used for training and 25% will be used for testing. So what that means here is that, you know, in the MSK data, the Sloan Kettering data, maybe 19 of these samples, these patients' data, 19 patients worth of data will be used for training, and then 5 will be used for evaluation. In one of the metastatic breast cancer, you'll have 7 used for training and then 2 used for evaluation. Because this SHAF data at the time was incredibly sensitive, and the Sloan Kettering data is still quite sensitive, we were only able to show very limited, you know, portions of the data. So in particular, we got permission to actually show 2 of the Sloan Kettering evaluation samples results and 2 of the HTAP. So that's what every figure will look like. These were both used for evaluation, not for testing. And the first thing that we show here is just a very blunt sanity check type of result. It's that if we look at a histology stain that is put into our model for testing, we generate a single cell data set from it. We compare that single cell data set to, you know, the goal, like what we're trying to generate. And we look, okay, how can we compare the bulk gene expression of these 2? This is more or less, you just take all the cells, you add them up, you find the mean expression value, and then just see how strong the correlation is. You can see that SHAF actually does decently well. This would be, what was I going to say? As you can see here on the left, it's very nice correlation. You see here on the right that like this kind of quantifies it, but more or less our baseline is also just a generative VAE. That's a type of generative model that is trained on the same training data as SHAF to generate single cell profiles. And, you know, that's going to be the common baseline because like this method is very unorthodox. There's not really a lot of baselines. So we have to be a little creative here, but you can see that SHAF pretty much outperforms the baseline very nicely here. And there's just a sanity check to make sure that, okay, this is, you know, this is working somewhat. Let's see if we can do more, you know, sophisticated type of evaluations. And the first thing then we can look at is let's look at correlations between different genes. So more or less, if you take our top row is just each of these evaluation samples. We have 4 that we're looking at here. You look at the single cell data set for each of these 4, and you see, okay, how strong is the correlation between, you know, gene X and gene Y in general. And you can get a correlation plot that looks very pretty. And then we can also try and do the same thing with the SHAF generated data. See like, okay, what are the different correlations? What do they look like? This is for 100 very highly variable genes that this plot represents. And you can see that SHAF, you know, it doesn't quite match it, but it resembles it far better than the baseline. Pretty much, it's nothing. But you know, it approaches it, which is very encouraging because it's a harder criteria. And then also we can look at UMAPs, and this is more or less asking the question of, you know, we try to visualize this data. At the moment, we don't really have spatial criteria. We do in a few slides, but not at this slide. And we'd like to just say, okay, we can visualize our single cell data, and how close are the visualizations between our original data and then what SHAF gives us. And you can see here is that, like, shape dynamics, color dynamics, clustering dynamics, qualitatively between the SHAF rendered data in the middle and the original are actually, somewhat encouraging. I like to think they are. At the bottom, at our baseline seems like the Italians say "ugaz," means nothing, and I like to think that. And then quantitatively, we could look to the right if we look at the silhouette coefficients, and more or less, this is just the strength of clustering of our different clusters. So you can see that obviously the SHAF data does better than the baseline here. Not quite as well as the original data, which are these very well clustered, of course, but still very encouraged. And that's kind of like more or less these past few slides are these non-spatial criteria. There's like, okay, we make sure our method works. And when we were developing this, all we had access to was the first corpus that I was discussing, the Sloan Kettering data, which did not have any sort of spatial transcriptomic data. We just had histology, and we had single cell RNA-seq. And then I'd say about a few months later, Aviv tells us about the Human Tumor Atlas project, which has those two things in addition to spatial transcriptomic data. Now, of course, heaven forbid we use spatial transcriptomic data in training, but we can use it for validation. It's wonderful. And because of that, the first question that you should come to mind is, okay, we generate our data, it's spatially resolved. How accurate is this, data? If we look at just this gene over space in the tissue, how close is it to, ground truth? So we fortunately have MERFISH data, which is spatial transcriptomic modality of, two of the samples that we're interested in here, the HTAP, two HTAP samples. And first, what we do is we look at 6 genes, and we'd like to just ask the question of, how strong is the correlation over space? When I say that, what we do more or less is we have a tissue, and we're going to just, again, using this window method, look at different spatial regions of the tissue, very small. I'd say roughly 30 pixels by 30 pixels, maybe a couple dozen micrometers by a couple dozen micrometers. And then look at the expression that SHAF tells us in that region, and then look at the expression that MERFISH, the ground truth, tells us in that region, and see how strong the correlation is. You could see here that we have, 0.5, 0.6 correlation for 6 genes. There's another more comprehensive figure in a slide or two that puts that into context, but encouraging that we're able to, not with a super strong correlation, but with a encouraging correlation, that SHAF's predictions are spatially sensical. And more or less, if we then look at, all the genes that MERFISH informs us about, it's on the order of a couple hundred, then what we're able to do is see, okay, how many genes have this strong of a correlation? Before I showed 6 that had a particularly strong correlation. What you even see here is that you have, scores of genes that are over 0.3, 0.4 correlation, which I know sounds not very strong. If you look at what else is going on in the field, though, that is comparatively very nice, though. So we're very satisfied to see this. And also then, what I think is probably the most, to me, is most aesthetically pleasing, but also is probably the most encouraging result is that we can look at a visualization of cell types. So I think this is probably a very nice summary panel right here. For instance, say that we have a histology stain that's seen in the top left, and we can put it through our model, and we get the spatially resolved transcriptomic data, and then we can do cell type labeling. I'm not going to go into the specifics of it, but there's just very basic automated cell type labeling that we can do. And then once we have that, you have something like we have on the far right corner, column, like you have just our tissue, and then it's color coded according to what the cell type is. And you might be thinking, oh, that looks very pretty. But here's the catch. Pathologists did the same thing. They looked at the histology stain, they said, okay, that region is this cell type, that region is that cell type. And these guys go to medical school for God knows how many years, do residency for many years after that, work in the clinic. And that guy said that, more or less these two align. So I take his word for it. And it's very nice. Again, you see like where there's supposed to be immune cells according to a pathologist. Immune cells, where there's supposed to be, vasculature type cells. You have vasculature type cells. So it's very encouraging. And we could do this for another tissue too. Well, it looks like it's a little bit prettier. If you ask me, this is like reminds me of a snake almost, this tissue, but closer up as to what the spatial visualization of cell types as predicted by SHAF look like. We do it for another tissue here. Not going to dwell on this for too long because more or less just a repetition of what we saw before. And this is still, though, with that being said, again, I'm biased here. I think this work is great. But with that being said, there's still some limitations. And the limitation here is that we like to think that, histology data is quite low dimensional, even though it gives, insights to morphological things at very high resolution. It's still only, RGB channels. And we'd like to maybe see if we can, upgrade our imaging. Maybe we can also, do a better job predicting transcriptomics. And this sets a stage for a lot of future work and work that's already done. So in particular, I'd like to give a shout out to, before I do that, I'd like to briefly introduce Raman spectroscopy, which more or less has been around in the physics community for a while. It was discovered by an Indian physicist named Raman back in the British colonial period there. It's been around for a very long time, but in biology, you don't really see it too much nowadays. So this is a very novel thing to maybe think, let's put Raman into a biological system and see what happens. And more or less what it gives you, though, is just like a greater insight into what the biomolecular composition of a sample is. It's very information rich. It's label free. You can use it to track live cells, which is the motivation for the work I'll be talking about 10 seconds. Very cool modality. And leads to, I would like to give a shout out to Koseki, who is not here at the moment, but Koseki is a postdoc in Peter So's lab. He's a postdoc at KCO, worked at the VIV, very strong collaborator with our lab. He's basically in our lab. Very smart guy, very nice guy. Developed this framework called Raman to RNA, which more or less is like SHAF, but is dealing with Raman spectroscopy instead of histology images. What is the difference here? Is that Raman spectroscopy is about a roughly 900 dimensional image, I put in quotation marks, but very, very high dimensional type of imaging modality that is more informative than histology stains in a lot of ways. And we just like to see, okay, can we go from Raman of, of a biological system to transcriptomics? And we were able to show, so for context, he's the first author. I am a contributing author, the second author, mostly took care of the machine learning and deep learning type tasks that we wanted to add for this. But more or less how we go about doing this is we look at a cell reprogramming system, iPSC cell reprogramming system, and see, similar criteria to as we discussed with SHAF, but just how well do we do in this system with these modalities as opposed to the others. And more or less just what we show through a lot of, I'm not going to spend too much time or dwell too much time on this, A, because it's I'm not the corresponding author, and B, we'd be here another few hours. But more or less, we provide a method for mapping, Raman spectra onto existing single cell gene expression data. So just if you have a sample of Raman spectra data, a sample of transcriptomic data, mapping one cell from one data set onto the other, we can also predict using deep learning. So using a similar method as in SHAF, this full adversarial autoencoder common latent space type setup, we can also translate from Raman spectra to gene expression profiles. And through a variety of criteria, again, won't go too much into depth because we're a little time limited. We show that we do a pretty good job though. And this is on the bioRxiv, I believe, right now. It's in submission. Hopefully it'll be published very soon. And yeah, again, with this being said, there are even still some limitations here. Requires, more or less that spatial transcriptomic anchor that I alluded to before. You kind of need this for this method. This is very expensive, and more or less just still, we like to think that we can do better in performance as always. And with this being said, this set the stage for future work. And future work here is we want to really consider just more new modalities, of course, new methods, of course, because I can't get a PhD without doing new methods. But we'd really like to consider more spectroscopy, like SRSs, John was alluding to before, other types of, very high dimensional, label-free, sophisticated spectroscopic modalities, see how much of a better job we can do with these as opposed to or in combination with traditional biomedical modalities. And then also we'd like to, explore, spatial transcriptomics. It's exploding. You see like, there's a graph somewhere. The cost goes down very quickly and the throughput goes up very quickly. And we'd like to see as that continues the next few years, just more or less how that can contribute to like these good methods and really showing that this task of connecting omics with images is not just feasible, but really promising. I was going to say that's about it for me. I would have a more thorough thank you slide, but I'm incredibly sleep deprived at the moment, mostly from insomnia. So I'd like to just say some thank yous. I was going to say my incredible mentor and PI, John, is here. My other incredible mentor and PI, Colin. Big thank you to him. Big thank you to friends that came out to watch me talk. Big thank you to Aviv for everything. She is incredible. And without her, without John, without Colin, none of this work would be possible. And yeah, thank you to the KCO, the Broad, MIT for funding, everything, so on and so forth. And that's about it for me. So this raises balls in your court now. Who has questions?